For generations, the origin story of the first Americans seemed largely settled. Ice Age hunters crossed from Siberia into North America over the Bering Land Bridge, spread south, and became the ancestors of indigenous peoples across the continent. The model was simple, intuitive, and taught as fact, but new genetic evidence is forcing scientists to reconsider that story. A major DNA study has revealed that some of the earliest Americans may not have come only from Siberia at all. Instead, part of their ancestry traces back to Ice Age populations living thousands of kilometers away along the northern coast of China. And the evidence for this surprising connection was hidden inside one of the rarest genetic lineages on Earth. The discovery comes from a large-scale genetic analysis published in Cell Reports. Researchers examined over 100,000 modern DNA samples and more than 15,000 ancient genomes from across Eurasia and the Americas. What they found challenges long-held assumptions about how and from where the first Americans arrived. At the center of the study is a specific form of ancient DNA known as mitochondrial DNA. Mitochondrial DNA, or mtDNA, differs from the DNA stored in the nucleus of our cells. While nuclear DNA is inherited from both parents, mitochondrial DNA is passed down exclusively through mothers. This makes it especially useful for tracing maternal ancestry deep into the past. Over thousands of years, mtDNA accumulates small mutations. These mutations form distinct genetic branches called haplogroups, which act like biological timestamps. By comparing these mutations across populations, scientists can reconstruct ancient migration routes and population splits. In this case, researchers focused on a rare mitochondrial haplogroup known as D4H. Today, haplogroup D4H is extremely uncommon, appearing in fewer than 1 in 200 people worldwide. But within this rare lineage are several subbranches that tell a remarkable migration story spanning East Asia, Japan, and the Americas. One branch, called D4H3A, is found among indigenous populations across the Americas and is considered a founding maternal lineage there. A closely related branch, D4H3B, appears primarily in East Asian populations, particularly in China and parts of Southeast Asia. This genetic relationship raised an obvious question. How could a maternal lineage central to Native American ancestry be so closely related to lineages originating far away in East Asia? To answer that, researchers reconstructed the evolutionary history of haplogroup D4H using advanced phylogenetic modeling. Their analysis traced the origin of D4H to northern coastal China, a region that includes modern provinces such as Shandong and Liaoning, along with areas near Beijing and Heilongjiang. From this region, D4H diversified into several sub-haplogroups. Some remained within China, others spread into Japan and Siberia, and one branch, D4H3A, ultimately reached the Americas. A key piece of evidence came from an ancient DNA sample known as NE5, recovered from the Amur River Valley and dated to approximately 14,000 years ago. Genetically, NE5 is only 6 mutations away from the Native American D4H3A lineage, making it the closest known Asian relative identified so far. This discovery provides a direct genetic bridge between East Asia and the earliest Americans. The study also identified 2 major periods when D4H lineages expanded outward from northern coastal China. The first occurred during the last glacial maximum, between roughly 26,000 and 19,000 years ago. This was the coldest phase of the Ice Age, when massive ice sheets covered much of the northern hemisphere and human populations faced extreme environmental pressure. Genetic dating suggests that around 22,000 years ago, a group carrying the D4H3A lineage began moving away from their ancestral homeland. These dates were calculated using a technique known as tip dating, which combines genetic mutation rates with radiocarbon-dated ancient samples. But this raises a critical problem. During much of this period, the traditional inland route through Beringia was blocked by enormous ice sheets. Travel across the interior would have been severely restricted, if not impossible. So, how did these people reach the Americas? The answer points to the Pacific coastline. Rather than crossing frozen land, researchers believe these early migrants followed a coastal route along the edge of the Pacific Ocean, a pathway known as the Kelp Highway. This route wasn't a single journey, but a series of gradual movements by small groups traveling along the coast. Kelp forests form one of the most productive marine ecosystems in the world, providing a reliable source of food such as fish, shellfish, and sea mammals. During the Ice Age, these ecosystems stretched continuously from East Asia to North America. This coastal corridor would have allowed humans to move southward without confronting the massive ice barriers that blocked inland travel, offering a plausible explanation for how people reached the Americas earlier than the interior corridor would allow. The second major expansion of D4H lineages occurred much later, during the last deglaciation between 19,000 and 11,500 years ago. As the climate warmed and ice sheets retreated, human populations expanded rapidly. During this period, new D4H branches spread across East Asia and into Japan. Lineages such as D4H1 and D4H4 became common in southern and central China, while others moved into the Japanese archipelago. These movements contributed to the ancestry of ancient Jomon populations and the modern Ainu people. At the time, land bridges still connected parts of China and Japan, allowing movement before rising sea levels submerged these routes around 12,000 years ago. Mitochondrial DNA tells only part of the story. To confirm their findings, researchers also examined paternal ancestry using Y-chromosome data. Here, they found a striking parallel. A Y-chromosome lineage known as C2A L1373, ancestral to Native American lineages such as CP39, was also traced back to northern coastal China. Analysis of more than 450,000 Y-chromosome samples showed the highest concentration of this lineage in the same region identified by the mitochondrial evidence. Two ancient individuals from the Songhua Plain, dated to around 14,000 years ago, carried both D4H3 mitochondrial DNA and the C2A L1373 Y chromosome. This strongly suggests that entire family groups, not just isolated individuals, contributed to the early peopling of the Americas. The genetic findings are echoed in the archaeological record. Across northern China, Japan, and the Americas, archaeologists have identified similar stone tool traditions dating back to the Ice Age. Stemmed projectile points discovered at sites such as Cooper's Ferry in Idaho, dated to around 16,000 years ago, closely resemble tools found at Jomon sites in Japan, dating to roughly 15,500 years ago. These tools differ from those typically associated with Siberian cultures, supporting the idea of cultural continuity along the Pacific coast, rather than across the continental interior. Additional evidence comes from ancient footprints, coastal settlements, and submerged archaeological sites now hidden beneath rising post-Ice Age sea levels. Taken together, these discoveries paint a far more complex picture of the peopling of the Americas. Siberia remains an important source of ancestry, but it was not the only one. Northern coastal China also contributed to the genetic makeup of some of the earliest Americans, supporting the idea that multiple migrations occurred along different routes and at different times. This research reshapes our understanding of Ice Age mobility, revealing that early humans were capable of long-distance coastal travel far earlier than once believed. It also highlights how much of our past remains hidden, and how powerful ancient DNA has become in uncovering stories that archaeology alone could never fully reveal. As more genetic material is analyzed and new sites are discovered, the story of the first Americans will continue to evolve. History, it turns out, is still being written. If you enjoyed this deep dive into how ancient DNA is changing what we thought we knew, consider subscribing to Hidden Strata. New discoveries are constantly reshaping our understanding of the past, and we'll be here to explore them.